Clostridium botulinum is a gram-positive bacilli, meaning it is rod-shaped and has a thick cell wall formed by multiple layers of peptidoglycan. As evidenced by these renderings, Clostridium botulinum does not have any evident cell structures such as flagella, pili, or fimbriae, and there are also no teichoic or lipoteichoic acids in its cell membrane. Clostridium botulinum is an obligate anaerobe, meaning it cannot survive in the presence of oxygen. This is because highly reactive molecules, called reactive oxygen species, form in the presence of oxygen, and they can be extremely destructive to cells. This is why nutritionists often recommend that we humans consume foods that are high in quote unquote antioxidants, which are molecules that can neutralize reactive oxygen species so they cannot cause damage to our cells. Aerobic bacteria evolved enzymes that, just like antioxidants do for us humans, can deactivate reactive oxygen species, allowing them to survive in the presence of oxygen. Obligate anaerobes, like Clostridium botulinum, do not possess these enzymes, so they're vulnerable to damage from reactive oxygen species, which is why they cannot survive in the presence of oxygen. Clostridium botulinum can survive temperature ranges from 10 to 48 degrees Celsius, growing optimally at 35 degrees Celsius, right around the normal body temperature of a human being. That said, Clostridium botulinum can form spores that can endure hours of boiling, so those spores would be classified as hyperthermophiles. In terms of pH, Clostridium botulinum can survive in pH ranges from 4.6 to 8.9, making them a quintessential nutriophile. Salinity, Clostridium botulinum can tolerate salinity of up to 5%, making them moderate halophiles. This means they can survive in water with no salt, to brackish water, to ocean water, giving them a diverse range of aquatic environments they can survive in. Since Clostridium botulinum is an obligate anaerobe, it does not undergo aerobic respiration. Thus, oxygen is not its final electron acceptor. Instead, Clostridium botulinum uses fermentation as its preferred method of metabolism. Fermentation starts with glycolysis, which yields 2 ATPs, 2 pyruvates, and 2 NADHs. So, pyruvate acts as the final electron acceptor, allowing NADH to be oxidized back into NAD+, so glycolysis can repeat itself, producing more ATPs for Clostridium botulinum to use. Based on the fact that Clostridium botulinum is an obligate anaerobe, it can only live in anoxic environments, meaning environments with no oxygen. In nature, for this reason, Clostridium botulinum is most commonly found in the soil, in bodies of water, and in marine sediments where dissolved oxygen from the water in the ocean cannot access the bacteria. So this just goes to show that Clostridium botulinum has an enormous range of habitats it can survive, since water, the oceans, and soil compose a huge majority of Earth. Clostridium botulinum does not have any motility structures, so it is moved around by its environment, such as waves or transferred on different organisms. In humans, direct ingestion of Clostridium botulinum cells does not cause infections because our stomach acid, which has a pH of 1.5 to 3.5, kills these neutrophilic bacteria. It is only when the cells enter the body through wounds or inhalation that they can establish infection. The case is different in infant human children because before 6 months of development, they do not have the acidic stomach profile necessary to kill Clostridium botulinum bacteria, which means they can establish infection inside the GI tract of infant humans, which is sort of an outlier but worth mentioning.